Hi, this is Will with MDPI Encyclopedia. Today we're delighted to welcome Associate Editor of the journal Livers, Professor Dr. Ralf Weiskirchen, to talk to us about his research on the use of immortalized cell lines in hepatology. So, welcome, Professor. Um, Before we begin, could you briefly introduce your academic background and your role as guest editor of Livers?
Hi, Will, and hi, everybody. I must say, I have studied biology some years ago at the university in Cologne in Germany. I studied biology and I make my PhD work in Cologne, ending in the year 1993. Thereafter, I have made a postdoc position at the university in Innsbruck in Austria. After working there for 6 years, I have returned to Germany, where I started my work on hepatic stellate cells, which is a special cell line relevant for liver fibrosis. And this work was conducted in the University Hospital of Aachen in Germany. And since that, I came back 1999 to Aachen. I work already 20 or 30 years in the same house. And my group is concentrating, as I said, on hepatic stellate cells, on liver fibrosis, and, for example, also cytokine signaling, animal experiments, and, of course, cells, characterization of cells relevant for biomedical research. So this is roughly what I do in my scientific career. And beside that, I also work for some journals, in particular for livers, for example, where I am the one of the associate editors. And I like this work very much because you have the chance to learn a lot about recent research, novel research, and you get good contacts to many, many people working in the field. So you have a good overlook at the complete field where I'm working in. So this is roughly maybe what I can tell the readers or listeners to our webinar or what it's podcast.
Cool, yeah.
Okay.
Thanks very much for walking us through your background there. So moving on to our first question then. What motivated your research into immortalized hepatic stellate cells, so HSCs, leading to the development and characterization of the GRX cell line?
So I must admit, in former times, I worked a lot with primary cells because primary cells are the gold standard in working in the field. But the things in Germany and also in the EU has dramatically changed because, we have a new animal welfare law and everything has changed. And we tried now to work models which don't rely on animals. So we are working with immortalized cell lines. They are easy to handle. They are much more cheaper than working with primary cells. You don't need a special equipment to work with these cells because usually if you want to isolate primary cells, you need special equipment, you need special machines, you need special reagents, which are very, very expensive. And so we decided to work on immortalized liver cells, particular hepatic stellate cells, hepatocytes, and we also work with some endothelial cells, which altogether are good animal replacement. So maybe you know that animal welfare has changed. We now must conduct the so-called 3R principle, which means that we should replace animals, reduce animal numbers, and also make refinement in research. So working with primary cells always rely on animals, which we have to sacrifice, we have to take out the cells. And if you don't need primary cells and you work with hepatic cells, which are immortalized, you can directly work infinitively with the cells you have in stock. So it's very manageable to work with these cells.
Okay. Excellent. Yeah, thanks for that answer. So, the GRX cell line is a key model for studying liver fibrosis. How does it overcome the limitations of primary HSCs or other immortalized lines, such as LX2?
Yeah, I must admit, in our laboratory, we handle many, many hepatic stellate cells from mouse, rat, and also from human. And what you mentioned, the GRX cells, was originally established in a laboratory in Brazil, in Rio de Janeiro, by Radovan Borovic, and he spread the cells all over the world. He had a special facility for that. He has a biobank who distributes the cells, and also the other cell lines are freely available, most of them. So you don't have to buy them. You can get them from cell banks, which is very, very cheap when you want to work with them. And if you want to study liver fibrosis, all these immortalized cells are good models, as I told you before. So they have some features which are essentially the same as in primary cells. They have, for example, a gene profession profile, which is very similar. They express a large of amounts of extracellular matrix components, which is a key in hepatic fibrosis. And they have also some functional features which also resemble that of primary cells. And last not but least, it should be said that when you have different stocks, different cell lines in your lab, and you repeat the experiments in different cell lines, you can overcome the problem which we have with primary cells. So you can work much more cheaper and much more reliable. Also, the planning is much more easier, because when you work with primary cells, you only can do experiments when you have them in culture. And to get them in culture, it might be a problem, because it's very, very difficult to isolate these cells in most laboratory. Also, we have in our laboratory established all the methods to do these studies.
Okay, excellent. So in your previous study, genetic and molecular characterization of GRX, what were the most surprising or clinically relevant findings about this cell line?
So there are many features of the cell lines which are remarkable. First of all, they have an expression profile which is closely resemble that of primary hepatic stellate cells. But usually, if you have primary cells in culture, they transdifferentiate from a quiescent phenotype, which is quiescent hepatic stellate cells. They transdifferentiate to myofibroblasts, which are totally different. During this transdifferentiation process, they lose their vitamin A. And in cell culture, the immortalized cell lines more resemble that phenotype. Usually, they don't have vitamin A stores. And what is remarkable in the cells is that these cells have still some vitamin A components and also genes which are relevant for the uptake of vitamin A. And they have a highly fibrotic activated potential, which means that the storage is quite good for vitamin A. And you can also add vitamin A into the cultures, and they take it up and they metabolize this vitamin A, retinoids, and so on. And so the subset of genes which are expressed in the cells are more or less the same as in primary cells. And this is remarkable. And on the other side, another remarkable feature is the genetic composition of the cell lines. So we have done genetic profiling, we have done karyotype analysis and also sky analysis. And what you see in this analysis is that GRX cells has a lot of rearrangements in the chromosomes. They have duplicates of chromosomes, they have losses of chromosomes. And what is remarkable is, even if they have these changes in the karyotype, they still resemble that of hepatic stellate cells, which is, I think, remarkable. So you don't see in culture a difference between the cells and primary cells. So this I found very, very remarkable.
Okay, wonderful. Thank you, Professor. So moving on to our next question then. So hepatic stellate cells play a dual role in fibrosis and regeneration. How has the GRX line advanced our understanding of these opposing functions?
Yes, as you said, hepatic stellate cells display a paradoxical nature. On one side, they're driving fibrotic scarring when chronically activated. They contribute to tissue repair. On the other side, in acute injury. So they have some good features and also some bad features. The good feature is in the setting of repair, and the bad feature is in the setting of fibrosis. So if GRX cells, you can culture them in different conditions, in different cell culture conditions, for example, with vitamin A or without vitamin A, and you partially can revert the cells to take a phenotype which resemble that of primary quiescent cells. So although they are fibrotic, you can reverse them into a more or less quiescent phenotype. And this is remarkable because this can be used in cell culture as a good model to make experiments, drug analysis. You can try if the drug is able to reverse the phenotype and vice versa. So this is a very good model. And these insights are invaluable designing for therapeutic interventions. So the chronic activation, because the demonstrating that you can reverse the cells from a diseased phenotype to a healthy phenotype or a more or less phenotype which is healthy, shows again that this model should be placed as a replacement for animal studies. So in the beginning.
Okay.
But nevertheless, at the end, you have to prove your results in animal models or in primary cells because, as I told you before in the beginning of my talk, it is the gold standard for doing experiments.
Okay, wonderful. So in terms of CRISPR now, so CRISPR and other genetic tools now allow precise modification of immortalized lines. So how do you envision engineered GRX derivatives shaping future liver research?
Yes, as you said, the adventure of CRISPR-Cas technology has opened doors for targeted knock-ins, for knockouts, and point mutations in GRX. So the researchers become able to precisely dissect individual genes and signaling pathways, which is once again wonderful for doing experiments. Because if you understand the pathways which are relevant for fibrosis, you exactly know what's going on, and then you can try to use drugs to compensate or to inhibit the relevant pathways. And the CRISPR-Cas technology has the capability to decipher fibrosis networks. You can look for genes which are relevant. If you knock them out and you see an impact on fibrosis, you know that this gene is relevant. And on the other side, you have also a capability to explore cell-cell interactions because hepatocytes and hepatic stellate cells, Kupfer cells, endothelial cells are all part of the liver. And if you only culture hepatic stellate cells, this might maybe add nothing to the complete knowledge of liver fibrosis because they are in vivo in crosstalk with other cells. And now you can look for specific markers which make this crosstalk. If you have a receptor which is able to recognize other cells, you can knock this receptor out, and then you can find if the interaction of the cells has changed or whatever. So it is very essential that CRISPR-Cas technology can prove the function of genes, and you also can add new genes into the cells, which then also has the possibility to clarify if a gene can act as an anti-fibrotic agent. So this is also relevant for fibrosis therapy. So to sum up, CRISPR-Cas is a good technology, and the manipulation of the cells with this technology will offer many avenues for establishing novel therapeutic avenues. So I believe we will learn in next years many things also on pathology of the liver by applying CRISPR-Cas technology.
Okay, excellent. So immortalized lines sometimes drift genetically over time. How do you ensure the stability and reproducibility of GRX in different labs?
This is a very good point. So when you compare cells in different labs, you often find differences, but this is due to the fact that they are not really 100% the same, because as you said, they become a genetic drift. So cells which are immortalized and you culture them for prolonged times, they have a genetic drift. They lose chromosomes, or chromosome parts of the chromosomes, and therefore it is really critical to use early passage stocks of the cell line. So prolonged culturing is not so good. So only use some passages, for example, up to passage 10 or 15 in cell culture. And it is also essential that you regularly monitor the cells. It can be the case that mycoplasm or something come into the culture, which has an impact on the gene expression profile of an immortalized cell lines. So it's essentially that you look for contaminants. And another very important thing, as I told you before, many cell lines are handled from one lab to the other. And it can be the case that there's a misauthentication or that something is not as it should be. And therefore, it's absolutely mandatory to test these cells, for example, by STR profiling, which is a technology, short tandem repeat profiling, which is like a fingerprint, a genetic fingerprint for a cell line. And if you look in the different laboratories, if the cells have the same fingerprint, you can be sure that the same are more or less the same. And what is very important or advisable, if somebody is working with a new cell line, it should be the case that he order the cell line from a cell repository, because this is essential. Because as you might note, one line in a laboratory is handled to a postdoc, from a postdoc to a technician, from a technician back to a PhD student, and they are usually put into different experiments. And the problem is that during this prolongation, maintaining of the cells, that you got a drift, that you got something in which should not be in, something goes out which should not become out. So it's very important at the beginning of the start to characterize the cells and be sure that the cells are really the cells which you want to work on.
Okay, excellent. So now moving on to our final question, Professor. So what advice would you give to young researchers selecting between primary cells, immortalized lines like GRX, or organoids for liver disease studies?
Yeah. So, as said 2 times, the gold standard are primary cells. But if you go into a laboratory, usually you do preliminary experiments. You first of all try if your constructs are okay, if your plasmids are okay, if a gene can express from a virus or whatever. So you only want to make functional analysis, not of the cells, but of your reagents. And therefore, it's very good to have immortalized cell lines, because you don't have to isolate primary cells. You don't have to sacrifice animals for your experiments. So you can do preliminary experiments all in immortalized cell lines, which is, by the way, much more cheaper than using primary cells. But at the beginning of the experiments, you will get only data, not knowledge about fibrosis per se. You only get knowledge about your reagents and your plasmids or whatever. If the experimental setting is okay, if the concentration of a component is okay. But at the end, when you do the final experiments, you must go back to primary cells, because gold standard primary cells are primary. Immortalized cells, the name says it, are tumorigenic in some cases. They grow, they have some genes which prevent the proliferation, maybe dysregulated. So at the end, you have to do your primary HSC. And the other thing are liver organoids, as you told us. This is a mechanism where you can, mechanistic aspect, where you can also save animal lives, and where you can capture different cells in the system where they form organoids, which are a kind of small organ. And this is also a very good thing where you can stimulate cell-cell interaction. And of course, you can also put GRX cells into these organoids and look how they behave. And you can compare them, of course, with primary cells. So I think it should be a good mixture of, in the beginning, of immortalized cells, then maybe organoids, and at the end, you need primary cells, and at the very end, you need animal experimentation. So if you have found a drug which acts antifibrotic in immortalized cell lines, you then go to hepatic stellate cells in primary culture, then you go to organoids, and at the very end, you go into the animal experiment. And if it's, an animal is different than a cell culture. In animal, they make bio conjugation, they take out the component, they excrete it, they metabolize the component. And in culture, everything which you put in will stay there. So there's no excretion. And so the concentration might be completely different. So these are the pitfalls which you have when you only work with cells. You have to translate your in vitro findings to in vivo experiments. And last but not least, this will require animals. But as I told you, you should always try to replace, reduce, or refine the experiments. So coming up to the end, maybe I would suggest, especially for persons which are not so experienced in cell culture, they first of all should learn all the techniques, and before they start with all these primary cells, they should be sure that nothing goes wrong when they work with primary cells. So this is my advice for younger persons working in the lab.